Old news by now, 25 years. But what I'll try to do is give you sort of the quirky in the trenches view, since I've been a scientist working on this for a long time, since the spill happened. And it might be a little eclectic, and I'm sort of saying things that you don't understand, just put your hand up and yell at me and I'll try to figure it out. And hopefully it's not just too technical mumbo jumbo. So the question is, what have we learned in all this time? And I'll just tip my hand right away. The important things to me are that the oil was more toxic than expected by a long ways. It remained longer in the sound than we expected, and consequently it caused more damage. I'll talk about the area that's affected. I'll talk about present conditions a bit. I'll talk quite a bit about toxicity studies. I'll use one to illustrate what's going on between us that do those kind of things, those type of studies, and what industry responds. And I'll talk about oil weathering. It's part of the package, and you have to understand that, and talk about what industry is saying. So the spill area was pretty large. It started in Prince William Sound, of course. It went on out through the Gulf of Alaska and got a lot of shoreline in the process. The Exxon Valdez spill was one of the largest in U.S. history and is considered one of the most damaging. About 11 million gallons of oil were spilled, and 1,300 miles of coastline were oiled. And it killed a lot of animals up front. We can go out today. This is 2012 data study we're doing out in the Gulf of Alaska on lingering oil, and you can find, you can dig out oiled rock from various beaches and look at the composition. So this is I'll talk about this a little bit more detail just shortly, but we have our lightest compounds here and heavier compounds going this direction. And we have a suite of compounds here that tend to be the toxic players in oil studies, still in this oil today. So just take a quick look at what the oil is like, what the composition of Exxon Valdez or Alaska North Slope crude oil is. There's a lot of aliphatic compounds in there. These are straight chain carbons, so you would have carbon, 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 all kind of lined up in a chain, if you can imagine that, with hydrogens bonded to them. The straight chains are about 5%. Other aliphatics where there is branching is about 80%. They're more or less straight chains. Then you have ring compounds, the aromatic compounds. These are the toxins in oil. And you have single ring compounds. They're often called the VTEX compounds. You guys familiar with this stuff or not? Yes? No? Some? Okay. And so these would be single ring compounds: benzene, toluene, ethylbenzene, xylene. About 6.5%. And the PAHs, not too many of them in the oil, but these are the toxic players. The interesting thing is, when the oil spilled and we had this film of oil spreading out over the water, these things were evaporating very, very rapidly. So within about a week or less, we lost almost all of these after the spill. So that left us with these toxins to think about and study. It was a paradigm shift for us to move, because we were studying this stuff. It's more water soluble, and more of it goes into the water. And so we're mostly ignoring this along with most of the rest of the world. But when this spill happened, we realized this stuff went away. We better look at this, and then we were in for a shock. And then there are about 7% other components. These include things like what we call the biomarkers, hoping sterins, those kind of things that are multi-ring and branch compounds that are even more complicated. This is what I'd like to show you is weathering in crude oil. But first let's talk about what these compounds are. Again, the molecular size is increasing in this direction in the diagram. And then this is be a relative percentage of how much stuff is there. The naphthalenes would be a 2-ring compound. Chlorines, dibenzothiophenes, and phenanthrenes is grouped here, 3-ring compounds. Some of them are called heterocyclics because they have an extra, different, instead of a hydrogen here or a carbon here in this case. This is a sulfur compound. And so molecular size is increasing here, and then we can have substitution, chemical substitution in these rings. So this ring here would be a C1 naphthalene. So that's illustrated as N1 on this graph. N2 would be 2 extra carbons, N3 would be 3 extra carbons hanging off these rings somewhere. So there could be a lot of different arrangements for these compounds to add to these, what's expressed here, summarized here. When the oil weathers, the relative concentration of the lightest compounds declines relative to the larger compounds. And I'll just run this. This is assembled from about 1,500 sediment samples collected in Prince William Sound and summarized in groups by what we call this particular weathering factor, which is just a convenient way of saying how weathered, how old is this oil. And now we've gotten to the other end of this scale here where we have relatively few naphthalenes and relatively large amounts of chrysines. So solubility is increasing to your left and persistence to your right. And the bad news is that toxicity also increases to the right. The more persistent compounds are the more toxic ones. So going back out here to our sample in the Gulf, we can also look at samples collected in the Gulf or collected in Prince William Sound and look at what I call the biomarkers, that other category. These are the hopanes, and we can ask whether the signal in this oil dug out of this beach matches the source oil, the Exxon Valdez oil, and in this case it matches essentially perfectly. And so we can use that kind of information to positively identify the oil throughout the sound today. This is an example of oil that's probably not showing too well in this light, dug out of oil reservoirs in Prince William Sound this year, thanks to Dave Jenker. And one of the things that struck us early on as we started working on this bill in 1989, of course, were these oil beaches everywhere, these ugly sites of oil beaches. And we got to thinking, this is an approximation here. This is based on mapping from Fish and Game, so that the oil would have kind of bathtubed around this stream delta like this. And we got to thinking, well, what happens when the tide comes in and it fills this area with water, and then the tide recedes, and that water, where does that water go, and what is it affected by this oil sitting here stranded on the rock? And the answer turned out to be, we actually did these studies later, in 2000, as you saw, that water drains into the stream, basically follows the topography. So if you've got a slope into the stream, it's going into the stream as the tide leaves. The groundwater will do that, and it'll pick up toxins from the oil and transfer them too, if that's what's going when that happens. And so we started thinking about, well, what can we emulate that in the lab? And so we developed what we call an oil rock column. Those columns can be fairly large. These are about 1 meter tall. These are maybe 20 centimeters. And the basic idea is that we boil that rock, much like you saw on the beaches, and we run water into these columns, and then we run it off and off to some experiment. And we started seeing embryo damage when we did that. This would be our control, pink salmon up here. This one is exposed to oil, and it should be pretty obvious that something is going on here. Edema, this large swelling and fluid retention is very symptomatic of exposure to crude oil.
Mark?
Yes.
Were the eggs then at the top of those meter tall
It
units?
depends on how we run the experiment. Some cases we've done it that way, some cases we run oil or the effluent water off to different chambers, depending on species or even different, we've done it different ways with the salmon. One of the points that we make in some of the papers is that we do have a physical separation between the salmon and contact with the rock, so that we can eliminate contact. It's direct contact is the transfer.
Yeah.
And you're flowing North Slope crude oil through that?
We're actually using North Slope crude oil. We pre-weather it in the lab. We remove about 20% of the volatile compounds, and then we apply it to the rock, and then we let the rock dry for about 24 hours, so that oil film it's stuck. It's not mobile at this point. Then we put the rock in the columns, and we usually run the columns another day or two with water flowing through to make sure there's no other oil particles that are going to move out of that column. So we precondition the column, pre-weathering it essentially, and then we start using it for gases. And we see this edema phenomenon and curved spines and other abnormalities over and over again when we expose animals to oil. So this is Pacific herring, this is zebrafish. These are recent. This is a 2014 paper, just came out. Bluefin tuna, yellowfin tuna, and amberjack, species of importance in the deep water horizon spill. And again, we get this gross edema happening relative to controls. And actually, it turns out that these observations extend back through time. I saw this in pollock, walleye pollock in 1982. I was seeing it earlier in my graduate work. Ole Linden was seeing it about that time too in Atlantic herring. So here are his controls, here are his oil exposed embryos, and Walt Pearson was seeing it with Pacific herring in the mid-80s. So we've known for a long time that oil is doing this. What we didn't know is exactly what concentrations were or just exactly how toxic this stuff was. Going back to our zebrafish, this is a crude oil exposure, and you can see using pure compounds, not much is going on with phenanthalines, but these three-ring compounds, the fluorines, dibenzothiophenes, and phenanthrines are causing havoc. So we started publishing in 1993. This is the first paper out of the block at our lab, and lo and behold, we felt that this experiment supported the mediation of oil movement or oil constituent movement, the PAHs, from oil to the embryo. And what we weren't really prepared for was this very, very low concentration, this very high toxicity, because we were seeing toxicity in the order of a part per million or so with our VTEX work, and then the PAHs start checking in almost 3 orders of magnitude lower. So we were quite startled about that. And in this first experiment, we're starting to see this evidence of edema in these fish. This work by Gary Marty in this particular experiment was pretty prescient, I think. This is a normal heart. Here's a heart in an oil-affected fish. So the edema is around the heart, and you can see this rather abnormal development in the tissue in the heart is abnormal compared to the control. This is an ongoing project we've been working on for quite some time. We're hoping to publish a new work on this pretty soon. The cardiac effect is quite profound and responsible for the edema that we see. The edema is actually a secondary effect as circulation fails, and the fact that the heart is negatively affected then reduces the potential for the animals to grow, and reduced growth is linked with reduced survival. We continued our work with this paper and found that, again, we confirmed that the PAH accumulation was mediated by aqueous transfer. We continue to find high toxicity, even higher toxicity, because now we're looking at around 1 part per billion in this paper. We started worrying about water quality standards and talking about that they should be improved, and we were really starting to see that weathering counts, because this 1 part per billion here was a single treatment within this experiment where the oil was considerably more weathered. It was actually oil that was reused from the previous year's experiment, and it checked in at a really toxic level, and we started wondering, well, why is that? We looked at it more detailed with this particular paper, which also, of course, was finding high toxicity, and the way we ran this experiment was we took a group of eggs and we exposed them to various oil doses, so that would be this gradient oil, this total pH gradient across the x-axis here, and then with a series of doses, then we get this nice toxic response curve. Then we used the very same oil rock generators, oil rock columns, and ran a second group of used them to expose a second group of eggs, and again we get this very nice toxic response in that group of animals. The interesting thing is those toxic layers those curves don't really overlay; they're displaced. So based on total initial pH concentration, we would say that the solution the first time was less toxic, and the solution the second time is more toxic, because we've got animals reacting at lower concentrations. So why is that? Let's look at, continue looking at the experiment a little bit more closely. The first group of eggs exposed to this less weathered oil was exposed to a series of concentrations, which illustrated in color here. And these concentrations declined over time. Pretty much, this is a pretty much linear, but this is a logarithmic scale, so this is an exponential decline in concentration over time. And the second experiment followed, and that decline continued to happen. So we have higher concentrations here, lower concentrations here in these two experiments. The first group of eggs accumulated PAHs over the 16-day exposure period, and then when we moved them to clean water, the PAHs started separating back out of that tissue. Whereas in the second experiment, the PAHs peaked at around 4 days, and then started degrading, and we then moved them to clean water again in 16 days. First group of eggs was exposed to relatively less weathered oil, so we have a fairly large naphthalene component here. Whereas in the second experiment, day 0, day 16, we have a relatively small naphthalene component compared to over here. So the two are looking at different weathers. So the driving force for such shifts in these toxicity curves can come from shifts in bioavailability, organism sensitivity, changes in mixture composition, and or the presence of unknown toxins. And so Neff here, in this past year, spent a lot of time thinking about confounding factors and whether they're significant. He reanalyzed and republished our experiment with this stuff, talking about microbial degradation, oil droplets, differences in stock, salinity issues, and other things. And so I thought I would talk about this a little bit as an illustration of what goes on, what has been going on, because we publish and then we get these sorts of responses, and it happens routinely. So this is not new for this particular paper. It's been repeated a number of times. So Neff analyzes our data and says, well, hey, look, you've got microbial degradation going on in there. Surely that must be confounding or must be adding to the toxicity of this study, and that must explain why, be one of the components that explains why these curves are shifted. And so he says, well, look, there's an initial microbial growth period here, and then this is getting established during the first part of the first experiment, and then the second experiment comes along, you've got a nice, healthy, robust microbial community which is munching hydrocarbons and producing metabolites, and they must be causing problems. This is C18 divided by phytates, and this is one of our straight chain alkanes divided by a branched alkane, and the branched alkane would be even more insoluble. C18 is highly insoluble. Phytane is even more insoluble, and phytane is less prone to microbial attack. So he's correct in that this response ratio could change. The problem is that we did not observe C18 in 60 to 64 samples in this experiment. So where did these numbers come from? These values were standard curves intercepts unrelated to the experiment equipment. How many people in here are familiar with standard curves in machinery? No? So what I should have probably should have put an illustration. The idea of whether you're calibrating a spectrophotometer or a gas chromatograph in this case, or some instrument, you want to know whatever that instrument response is. You want to be able to translate that back into numbers that you understand in terms of the chemistry, in terms of the concentration. And so you feed the machine essentially a series of concentrations, and then you use a standard regression to say, okay, how do these concentrations relate? And so you want to, of course, you want to have a really good tight regression. So there's a lot of thought put into some of this kind of stuff. And then once you understand, once you've run that standard curve, you can use that information to run the regression backwards and say, okay, now I've got my unknown produced this value, what is the concentration? So NEF actually analyzed the standard curve intercepts, which were inadvertently published in the database at that time. We corrected that problem. And there was a series of numbers that looked like this, which he should have understood was a problem. So the experiment does not provide support for microbial toxicity. This experiment or any of them produced. He talks about oil droplets as being present and confined. This has been an argument, a technical argument between us for a long, long time. And I suppose one of the easy responses here is that the C18, which is highly insoluble, it's absent. I just demonstrated that. And but we could take a closer look at the experiment itself and see how this behaves. So here's our oil drop column, the loading on it. So the amount of oil that we stick on the column is increasing to your right, like that. And when that happens, the toxin that we put in the water, the pHs, increases. Meanwhile, these insoluble alkanes, which would be considered a tracer of whole oil, are doing nothing. And you can see the same pattern happening, a pattern happening with pHs, the toxins, over time within the experiment. Notice I've got a linear scale now, so your exponential curve looks like it's exponential. And still nothing's going on with the alkanes. When you turn to the embryonic tissue, again you see things happening. You see uptake with the PAHs, the toxin, and you see nothing going on with the highly insoluble stuff, the highly insoluble alkanes. So you would think probably there's no whole oil present, and indeed when you compare the whole oil alkane composition, which this is in whole oil here, to what you see in the embryo tissue, there's no relationship. And the same thing with water. Again, here's your whole oil composition, and that is not what's going on in the water. That's really different from your pH, where what's going on in the water, here in orange, or in the tissue in orange, is the same as what was in the original contaminating oil. like I've been foiled by my computer here a little bit. Does this technique actually work? You know, when you start doing an analysis like this, you worry about, well, you know, I just told you a story, or is it real? Can I actually detect particles? And we turn to what we call a water-propagated fraction experiment, where we've taken oil and water and physically mixed it together vigorously, which produces all kinds of particles. Can you see those particles using this analysis method? And the answer is yes. So here's our PAH toxin increasing with loading, and now our alkane is increasing with loading too, signaling the presence of oil droplets. And not only that, but then the composition in the water looks like what's in the oil. When you turn it to animals exposed to this very same mixture that has oil droplets in it, we again fail to see alkanes responding in some comprehensible way. It goes way down here, it comes way back, you know, what's with that? I mean, it's just wandering around. And there's no relationship between, there's no petrogenic oil-based alkanes going into these animals. So even when they're exposed to oil droplets, you don't pick them up. So you start thinking, well, oil droplets can't be too effective. We did another experiment to look at this particular problem. This was published back in 2008. It was a rather elegant experiment, and I can say it's elegant because I didn't think of the really elegant part of the experiment, my colleague did. We embedded a group of embryos in augurose, or an auger, and that would allow anything that was soluble to interact with the embryos, but it would prevent particles, oil particles, which we overlaid on top of this petri dish, from interacting with these embryos. And then we also paired in every single concentration dish, we added a bunch of embryos up here so they were exposed to the droplets. Here's our embryo, example embryo exposed with oil droplets, and here's one where it's isolated from droplets. And if you analyze the data and figure out what the concentration, what the dissolved concentration only in the auger layer, and the dissolved concentration only in the overlying oil droplet laden area, then these curves basically tell you the same thing. This is edema happening, and it happened at the same rate in these animals. If you were to ignore the fact that some of these are actually in droplet form and measure all their oil, plot this against all the oil in the experiment, then you will conclude that droplets, the oil is a lot less toxic than you do looking at it as a dissolved fraction. These sorts of arguments just sort of seem to go on and on. I'll step through this quickly. It seems like, you know, there's a lot of stuff being thrown at us in a hurry. So, what's valid or not in that, I'll let you think about. We talk about stock differences in this particular experiment as being important because we got our animals from the source over here in one case, and over there in another. They're both from southeast somewhere, I don't remember exactly where. And I point to the literature and say, okay, here are a series of various species, mixed species of embryos that have responded negatively to oil, to pHs, total pHs in some way. And so here I've got up to 20 parts per billion here, down to 10 parts per billion here, down to 2 parts per billion here. We still have a bunch of species that are reacting negatively to. And so we don't know where this is going to asymptote or whether there's threshold out here, but it's a small number, and it involves a number of species. So I think the stock argument is not too convincing.
Mark, again,
Yeah.
The discussion and the NEF is that I didn't get right off the start. Was that?
NEF would be an oil representative.
Right. I wasn't sure if I wasn't catching that, and I wasn't sure if others were. from your main study, then you're getting knocked at by NAF, and then you're
Right.
shown, by the oil industry.
Right, and so he's
And
one
then-
of several.
And then you're showing the corresponding response to his criticism, showing
Right.
it's
Right.
not correct. Right.
Correct. Another criticism that came in on this experiment is non-optimal salinity. I found that one quite puzzling. We're pretty good at handling, at rearing larvae, or rearing embryos to larvae. We get good viability, good hatch, good viability with our controls, and we did an entire study looking at this with thousands and thousands of animals. This was published by Johnson in our lab. So we're good at that. Salinity was the same between experiments. Why do these things come up? Incidents of abnormalities in the field, irrelevant. We have good controls in multiple experiments. We've done many experiments at the lab. We're getting good controls. We're getting good dose response results in multiple experiments. And Neff participated in this study with Brandon et al. I saw the same thing with Big Salmon we did. They obscured the entire study in an interesting way, but they saw the same thing. So we conclude that PAH exposures lead to the same types of embryo damage regardless of dosing methodology, timescales, water sources, and species. And I guess I should highlight in that in red. The dosing methodology, whether we use the oiled rock columns, which seem to routinely come under attack, or whether we mix the oil and water in a blender, making a waft, or whether we use some sort of partition-controlled method, it all does the same thing. If you're moving PAHs into the water where they're available, biologically available to the animal, they're going to cause damage. The time scales are very, very different, which tends to eliminate arguments like the microbial development, microbial metabolites. Microbial communities take a while to get to grow and establish themselves and do something. And so in the zebrafish experiments, we're seeing responses in the order of hours to days because these are high temperature, rapidly developing embryos. We see responses in herring embryos in days to weeks, in colder water species. In the pink salmon, it's weeks to months, and we see the same thing going on. Water sources include freshwater, saltwater, special system water, which basically starts with deionized water. Still the same thing, and as I showed you earlier, it happens with multiple species over and over again. So exposure to oil is consistently damaging, and PAHs are the most likely toxins in that oil. This is not only our conclusion, but this is a conclusion people have made around the globe over and over again for several decades. And industry has to agree with that. But then they also talk about our experiment as not reliable and not producing reliable results relevant to the field. So one of the things I learned during this spill, and I wasn't prepared for this, was that for every paper there is an opposite anti-paper. Actually, there are quite a few opposite anti-papers. The industry tends to do things that you have to pay attention to when you read their work. This one, for example, the statement is distinguishing between the oil spill impact and everything else was challenging. And so this is sort of an illustration of Prince William Sound, and they talk about villages and mining and canneries and all this stuff that goes on. You got fishing and commercial boat traffic. Things might be coming in from the atmosphere. They talk about forest vegetation and seep oil coming from the eastern Gulf of Alaska. I have to remind these colleagues that the Exxon Valdez oil spill exceeded the hydrocarbon input into the sound by a long, long ways. Less than 0.2% of the Prince William Sound shoreline was contaminated by human activity. So we'd probably eliminate that. 14% was contaminated by Exxon Valdez oil. Coal was the most likely source, is the most likely source of hydrocarbons entering from outside the sound. It's coming in as ground particulate load in the water. It doesn't change, it doesn't weather, which means that whatever those PAHs are locked up in a matrix that's immune to weathering. And so this is not a plausible hypothesis. We've spent quite a bit of gas writing papers on that. Fires and vegetation, that's pretty easy to eliminate because you can see pyrogenic signal very clearly in the chemistry. So these arguments are not really holding too well. Industry perspective is that weather remnants are a minute fraction of the total shoreline. That's actually true. It's there's not, in terms of the total shoreline, there's not a whole lot left. But what we do have to worry about is what is left is still quite a quantity, and what has it been doing all this time?
How much would you say quite a bit quantity is?
Well, I did some back of the envelope calculations, and so there was about 14% of the shoreline, 15% of the shoreline oiled, and at the time, a recent paper that we put out said, well, there's about 14% of that oil remaining. So we're down to about 2%, still about one order of magnitude greater than the estimate for things like the canneries and past history in the Sound.
But for the Sound, that one paper was about, what, 18,000 gallons?
I don't remember what the number is, the short paper.
Yeah,
Yeah.
in Prince William Sound,
Yeah, there's a substantial amount of
Yeah.
oil there. Even though we're
still
talking
under the
about-
sediments, and that's just in the Sound, not the Gulf.
So the question is, it's discoverable. We can discover it when we do surveys. And the problem is that otters have been discovering it too, apparently. And I'll get into that in a minute. Industry talks about the wildlife recovery being fairly rapid, and talks about CYP1A. This is cytochrome P451A. That's a big fancy name. It's a biochemical marker that we use to understand oil exposure in animals. We can use it in fish. We can use it in otters. It signals the presence of planar hydrocarbons such as the PAHs. It also can signal the presence of other things like persistent organic pollutants if they happen to have that type of structure. Recovery time, I've kind of sketched out some recovery times for various species around the Sound. Pink salmon probably took about 4 years before that chronic exposure was depleted to the point where it wasn't really effective anymore. Mussels, again, a chronic exposure issue for about 10 years before concentrations in the underlying sediment dropped sufficiently to that we weren't really getting biological exposure in this species anymore. Harlequin ducks and sea otters, it's taken more like a quarter century, more than 20 years before and again, this is a chronic exposure issue with encounters. Oiled sediment, that's going to be with us a while, 50 years maybe more. The killer whale AT1 pod is a different story. These animals have suffered an acute exposure and it knocked out the matriarchs. And so this pod will probably never recover.
Why do you show the, oh, never mind.
The herring's an interesting story because there's controversy around there, and we wonder, is it worked as an oil exposure problem or not? And assuming it's linked to the spill, then the herring story is still going on too. And
Do you think it's like this, Bill?
do I think it's linked to the spill? I've struggled with that for years. And in the paper that I put out, the synthesis I put out on that topic, I said we left the door open. There is no obvious direct link, but we all wonder and we all think that
I was
You are
out there
coming
in
up
the
with some-
spill, and I
what's that?
saw herring jumping through the oil out there in the spill,
Mhm.
flopping down on it, and it was so thick, they just, wobbled a little bit.
What did they do after after that
Well,
happened?
we never saw any dead herring.
They just disappeared.
Yeah, they were flopping through the oil trying to breathe.
Mm-hmm.
There were a lot of them. It was, very time.
This business of impaired hearts may be part of this story, which pushes the idea that the spill had an influence in the direction of causing the problem. But whether the spill caused the problem or not, what is really obvious today is that you don't just fix an ecosystem. Once they're gone, it's a long-term problem that we humans really don't fix. We say we manage them and we stop the fishing, but don't know. They're not rebuilding in a way that we might predict it. It's just by simply removing fishing pressure. The sea otter story is looking pretty bright. The North Knight Island population is returning to preschool abundance. Mortality patterns are normal, so we're no longer seeing death in the prime adults. The gene transcription rates are looking okay, and this is suggesting a gradual dissipation of the oil, a gradual decline in their encounter rates, and that this is no longer of significance, or the significance is being reduced. So that's good news, but it took a long time. And so this is a part of the story of a long time, much longer than anyone would have expected would have happened, especially with a species like this. A teapot again is probably going into senescence. Industry tells us the remaining oil is highly weathered and therefore not toxic. That's just flat out not true. Here's our example again from the Gulf of Alaska, where we have plenty of the three-ring toxins still remaining. They talk about remaining oil being in the uppermost intertidal zone. We go out and we look, and know it's distributed through the tidal zone. Here's low water here, up to the upper zone, and so we've got some sort of distribution in the subsurface oil in that zone. So this argument is convenient because it says, well, it's most of the oil is removed, most animals are removed from exposure to oil, but it turns out that's not really true. And then, of course, we've had people working back with the otters and estimating, how often would these individuals encounter oil during the year. And so that looks like it might be in the range of 5 to 10 encounters per year on average, or median.
Has the industry come out with a study to mimic your studies to either prove or disprove your claims?
In the case of pink salmon, they produced a study that was essentially identical. It was modeled exactly after our stuff, and what they did was they related all effects back to the amount of oil stuck to the rock. So that's sort of a nominal oil, and we put on thousands of parts per million oil on the rock, and so our animals responded to thousands of parts per million. They did the chemistry, they reported the chemistry, and they got the same sorts of results with it. And they had animals that were not in contact with the rock, they were in contact with water that was laden with less than 8 parts per billion PAHs, and those animals responded negatively. They were damaged. And so they say they can't replicate our results. In fact, they did. So it's an interesting mess to sort out. So I would conclude that Exxon's oil was more toxic than expected. It caused longer-term damage than expected, and it caused more damage than expected. I also learned something I didn't want to know, and there's an anti-paper for every paper. Lessons that we really ought to pay attention to are: be prepared, be watchful. Don't just rely on the government to get it right. Don't rely on industry to get it right. We all need to be involved and pay attention to what's going on. Unfortunately, oil spills seem to be inevitable. They seem to be happening with discouraging regularity. Cleanup is basically impossible. Very little of the Exxon spill was recovered, even after all that money was thrown at it and all that time. Restoration is impossible. We have no idea how to fix an ecosystem. Prevention is the key. We gotta figure out how to stop the spills, and one of the ways to do that is to stop burning hydrocarbons for energy and move off to a sustainable energy that won't cause these kind of problems. That's a big, tricky, huge problem for society. And that's my rant for the night.
Do you think Exxon Valdez was one of the most devastating because the industry was not prepared? They didn't have the tools in place. They didn't have plans in place, and they more or less kind of pushed that to the side. You know, put it in the back closet.
That's clearly the problem. And you know, I don't know. You know, looking 20/20 hindsight, I'd like to say, well, yeah, they could have contained it in the first 3 days before the storm blew up, pushed it all over. Whether they could have actually done that or not, I don't know.
I look at it as a perspective because I work for industry on oil spill response plans, emergency response plans, and I also do some contract emergency management planning for a few industry corporations. And I look at oil spill now, and I've heard the way Exxon was handled and the way industry is looking at it now, you know, is tenfold, you know, from where they were to where they're at now, because of the awareness.
That's clearly an improvement.
And you know, I think that the Exxon, the toxicity levels could have gone down if they started doing, you know, if they could burn, they did the institute burning right off the bat, you know, but now does this cause another environmental, you know, if you're trying to get rid of one chemical and you're burning it, yet now you're releasing it in the airway. So it's like, pick your poison, which one do you really want?
You are absolutely right about that. And so another big debate is the dispersant use.
Yes.
It's the same issue. You put dispersants, apply dispersants, and then you're going to have a lot more of this stuff in the water column, where you've got water column animals being exposed, or do you let it go off on the beach?
Yes.
Now, of course, people that like water column animals might say, well, we want this, we don't want this. People that say, well, you know, it sticks on the beach and causes longer-term damage, ultimately, we don't want that. And so those kind of debates are very legitimate.
Yeah, and I see corporations and companies that have taken the stance, and like you said, disbursements. You know, I had an active role in the Deepwater Horizon through an oil company that I was working for up on the North Slope. Went down there, and the argument of disbursements, and what they were finding was the deoxygenated level in the water. You know, that oil was sinking after they dispersed it. Where did the oil go? Well, that's the magic question, and where's it going to end up? So you're trying to remove one toxin, you know, so now you're adding another toxin. So which poison do you want?
The two together aren't.
The two together aren't great. And like you said, oil spills are inevitable. You know, it's going to happen. You know, we as a country rely. You have to have petroleum, regardless of what type of energy source you do. We go to, you know, wind power. Those engines have to be lubricated, which is petroleum. You know, so it's like.
But if we're not burning petroleum,
Yeah.
we're using it for medical, or we're using
Yeah.
it for plastics, or we're using it for lubricants, we should be able to reduce the amount that we consume by oil.
Yeah. Oh, I fully agree, and you know, I've seen what oil spills do. You know, I've worked on the North Slope where I've watched oil spills ruin the tundra, but the only reason it ruined the tundra is based on the response they had with the tundra.
that could be a problem too.
So I see industry taking a more hardline stance towards prevention, especially after the Deepwater Horizon, where BP tried to push the buck, and they're still paying for it, and they're still held up in court over it. And the finger pointing's got to stop, you know. For every finger you point out, there's a thumb pointing right back at you, you know. So,
Now, there's a, you know, at the Exxon Valdez, it's like you can't get a street light at a dangerous corner until somebody's killed there. So at the Exxon Valdez, you ended up having a real disaster take place that then initiated, you know, a lot more response and training and
exactly.
oversight and crew need, you know, the crew, the human element aspects. So all those things came into place. It's still not enough, obviously, but it is better.
It's just like any occupational safety hazard, you know. It takes an injury or an illness or a fatality for the standard or the regulation to change,
Right.
for us to learn. And, you know, it's,
And some of this has it, is the
Yeah.
thing too. The
Well, that's-
Clean
that's
Water
what-
Act is part per billion, and the
Right,
stuff that
that's
they're
what we
coming out
have
with
to-
is a part per billion,
That's what
and that hasn't changed.
we worry about those arguments is because we see things, you know, that we know are highly toxic, and we want to win the argument because we think it's better for society.
So when you did your studies of letting the embryos, did you let the embryos get to adulthood, or did you end up?
I didn't show you any studies like that, but we did do some studies like that, and it was very interesting. Pink salmon are salmon in general, but pink salmon are very helpful because you can rear them in the lab. You obtain them from wild stock that came in and basically handed you the eggs, so to speak.
Yeah.
And you can rear these eggs in the lab and then say, okay, on out into the North Pacific and do your thing. And they come back.
Yeah.
And when they come back, the oiled ones come back, fewer oiled ones come back, relatively fewer oiled ones come back. And so there was a statistically significant reduction. We had 3 different oil levels. We actually repeated the experiment several times, and it happened. The same thing happened every time. We would always get relatively less oiled animals back. So they weren't surviving as well. And the reasons we think they weren't surviving as well is because they did not grow as well. We could do experiments 3, 6, and 9 months after the experiment and watch how fast are these animals growing, and they're growing slow. So, and growth is an important survival strategy for these animals in the wild. And the reason we think they're not growing as well is because even when they look normal, we're still seeing evidence that those hearts were affected by pH molecules early on, and we think they're in the tricyclic range. And so those hearts are slightly changed from what they are. So they have reduced aerobic capacity, which means they have reduced potential to grow. And so there's this lifelong impact on the affected individual.
I have a bunch of questions, but one would be, the figure 11 million, I've heard that, but I've also heard other people say it could be as much as 30 million.
I don't know what the true answer is. It's contested.
Okay.
The official line is 11 million. It could be more.
So it could have been,
I don't
I've
really
heard
have any
30
good
million.
information on that. Other than
Nobody does.
I'm aware that
Yeah. My other question was, your findings, is anyone using that as evidence to go back to Exxon and to talk to them about, well, it's actually not as recovered as you think, and is anybody doing anything with all
One of the
this?
reasons why there's so much controversy in the literature today is because the reopener clause is sitting in the courts.
Right.
And the courts at some point are going to have to decide, and the debate is going to happen, I think, in the courts, as to what all the stuff that we produced, McCullough's produced, whoever's produced it, on both sides of the fence, and try to sort out what is the truth, what is, how much damage was there more than originally settled on. So that's a live issue that's effective is.
And a couple of questions that are sort of related. I was wondering if the PAHs travel up the food chain as these fish get preyed upon.
The answer is no. That's good news, actually.
That is
Persistent organic pollutants, things like DDT, which are basically biologically impossible to grind up, will accumulate in the food chain. And so the upper level predators get a higher and higher load as you move up through the food chain. The PAHs are ground up. The biology of the cell, or the biology of an animal, the biology of us, allows that those molecules to be metabolized. And that CYP1A stuff that I was talking about, that cytochrome P450 enzyme system is induced by these molecules, and then it starts degrading. So it's degraded by that on cellular level. Microbes will attack both the straight chain alkanes, the branched alkanes attack. Eventually they'll attack the ring compounds and start oxygenating so that they eventually it breaks down. It doesn't persist the same way.
That actually answers my second question as well, which was if they, like the pink salmon you were talking about, did come back, if they would pass those on to their young as well. But if it gets metabolized, then obviously it wouldn't pass on directly to the young.
Right, and I think we see the hydrocarbon load come out so fast early on that by the time those animals have been out in the wild and then come back as mature adults, the load would be insignificant, I would think.
So all the damage is done up front.
The damage is done up front.
Do you think?
I'm so sorry. No, you go for it. I can remember that.
Do you think some of the coastline that's still affected is just based on the cleanup activities, that cleanup was not able to reach certain coastlines, so oil remained there because it was not cleaned up?
Well, there may be some of that, but it's also pretty clear that even places that were cleaned up are still affected. So our group, for example, said, okay, let's do an experimental cleanup, and we went to mussel beds that were oiled, and I can't tell you whether those were cleaned up during the official cleanup or not. I think they were, but then we went back later and said, well, there's still problems here with these beds, and we took the mussels off, we set them aside. We're talking about fairly large sections of beach, and then we scooped off a layer of oiled sediment and put that aside, and we filled in other sediment back on the bed, put the mussels back, and then sat back and watched. And somehow the oil that we didn't get, which was even deeper, started showing up in that layer, that replacement layer. So you can see those kind of things happening. It's very, very difficult to do this stuff. So you've got that sort of thing going on, and what we didn't anticipate was in these high energy environments, particularly out in the Gulf of Alaska, which some of these shorelines just get beat, but they're armored with rock, and that oil manages to sit under those. It's stable enough that the oil manages to sit under that stuff and stay. So it's fairly well insulated from the environment. What you do worry about then is the big storm events that are going to turn some over and then have biological availability, or something like an otter that appeared to be, was digging into the sediment and encountering oil and actually excavating itself.
How much would you say or guesstimate that is Exxon Valdez oil versus natural seeping oil?
It's almost 100% Exxon Valdez oil. And I'm setting on a retrospective right now in the lab. I have samples across time in the sun, so I'm doing an analysis. We're now analyzing these biomarkers that we weren't looking at initially during this early years. We started that only 3 years ago. And so I'll go back and relook at a bunch of these samples, and I'm quite sure that we will see every time Exxon Valdez oil signal as we go back in time, because they're highly conserved, and they're highly refractory, they're highly persistent, and they're very good indicators, accurate indicators of the source. So eventually we'll have that information, I think, very clearly. In the meantime, we do have other experiments that all point that direction. That answer the question already?
I was gonna say, when you had your oiled salmon and they returned, did you do a study to see if they're still fertile or not?
I think there was, I'm not familiar with that, I think we had one experiment that we looked into genetics in that fashion, but I'm not familiar with that experiment. Okay.
Thanks.
All right, thank you.
Who's your biggest industry opponent that's trying to argue these papers?
Wait, there are a number of researchers that go ahead and look at this.
Yeah, we'll shut the video camera off when I tell you.